Imagine for a second you have a ball of modeling clay sitting on your desk.
Okay.
You can squish it, you can roll it, maybe you can poke a hole in it. But if you walk away and leave it alone, it's just going to sit there.
Right.
It is not going to suddenly decide to organize itself into a horse or a starship or, a fruit fly.
Unless, of course, that ball of clay is a living embryo, in which case it does exactly that.
Exactly.
It doesn't just sit there. It computes. It self-organizes.
And that is the central mystery we are tackling today, the embryo problem. How on earth does a chaotic ball of effectively identical cells spontaneously organize itself into a complex functioning animal?
I mean, it's the fundamental question of developmental biology.
It feels like it breaks the laws of entropy sometimes.
It really does. And to answer it, we are doing a deep dive into a groundbreaking paper published in Nature Methods titled MultiCell, Geometric Learning in Multicellular Development.
This comes from a collaborative team, MIT, University of Michigan, Northeastern. And their mission, which is now our mission, is to explain a new AI framework called MultiCell.
Right.
But I want to be clear right off the bat. This isn't just another AI looks at pictures deep dive. We aren't just categorizing images here.
No, not at all. This is about predicting the future of living tissue, cell by cell, using geometry. They've built a framework that explains every mechanism and biological implication of how cells move.
And we are going to unpack this from the ground up.
We are.
So before we get to the neural networks and the fancy math, we need to understand the terrain. I know in this field, there's been a bit of a historical feud, right, regarding how to even look at a tissue.
A conflict of perspective is a polite way to put it. Historically, if you want to model a tissue to understand how it grows, you had to pick a side.
Pick a side.
Yeah. Camp A was the granular picture.
The granular picture, that sounds like particle physics.
It is, essentially. You view the tissue as a point cloud. Imagine a ball pit at a playground. Every cell is a plastic ball. They can bang into each other, move around, and you can track them individually. This is fantastic for, say, tracking gene expression or distinct cell movements.
Right, so the granular view treats cells as independent agents. But cells aren't floating in a vacuum.
No.
They're stuck together. I mean, if I pull on my skin, the whole sheet moves, not just one particle.
Exactly. And that brings us to Camp B, the foam-like picture.
Right.
Think of soap bubbles in your sink. When you look at bubbles, you don't really see the balls. You see the network of walls where they touch. You see the surface tension.
Right. If one bubble pops or shifts, it instantly pulls on the walls of its neighbors. It's like a mechanical web.
Precisely. That's the foam view. It's excellent for understanding forces, stress, mechanics. But for decades, these were separate mathematical worlds.
So you had to choose.
You either modeled the balls, the cells, or you modeled the foam, the junctions. You couldn't easily do both because the math just didn't play nice together.
And the breakthrough of MultiCell is that it refuses to choose.
It refuses to choose. It says, we need both. And that leads us to section 1, the mechanics, the dual graph architecture.
Okay, let's visualize this because dual graph sounds pretty abstract. How do you actually feed a living, squishy embryo into a rigid computer system using this method?
You digitize it by layering 2 maps on top of each other. This is essentially equation 1 in the paper.
2 maps.
Graph number 1 is the cell-cell graph. In this map, every node is a cell. The lines connecting them just represent adjacency. I am sitting next to you.
Okay, so that's the granular view, a social network of cells. Who is friends with whom?
You got it. But simultaneously, the model builds graph number 2, the vertex edge graph.
Okay.
Here, the nodes aren't the cells. The nodes are the vertices, the tiny corners where 3 or 4 cells meet, and the edges connecting them are the actual physical junctions, the membranes.
So to use a city analogy, one map shows just the houses, that's the cell-cell graph.
Yes.
Yeah, the other map shows just the intersections and the roads, that's the vertex edge graph.
That is a perfect analogy. And if you want to understand traffic flow, you can't just look at the houses. You need to know if the intersection is blocked. You need to know if the road is narrow. You need both maps.
But a mat is useless without data. This brings us to the input features. And I'm looking at the list in equation 3, and it's extensive. They aren't just feeding in location X and Y.
No, absolutely not. Raw coordinates are actually not that useful because embryos move and rotate. They feed in what are called invariant quantities.
Invariant.
So for the cells, the AI measures area and perimeter classic geometry. But it also measures Gaussian curvature and mean curvature.
Okay, I remember Gaussian curvature from calculus, vaguely. But why does a single cell care about curvature?
Well, Because embryos aren't flat sheets. They fold, they invaginate. Gaussian curvature tells the cell if it's sitting on a hill, a valley, or a saddle shape. It's a proxy for the 3D stress the cell is under.
Ah, so if you're being bent, your curvature changes.
Precisely.
Okay, so the cell knows if it's being bent. What about the roads, the edges?
This gets really interesting. They measure the edge length, of course, but they also measure something called the Delaunay area and Delaunay shear.
Shear usually implies tearing or sliding, like scissors.
And in this context, it's a measure of distortion. Imagine drawing a triangle connecting the centers of 3 neighboring cells.
Right.
If those cells try to slide past each other, that triangle stretches or skews. That skew is the shear. It tells the model how much the tissue is twisting locally.
So we have the shape of the house, which is the cell, the shape of the roads, the edges, and the twist of the land, the shear.
And one more critical thing. The model calculates the rate of change for every single one of those numbers.
Oh, wow.
It's not looking at a photo, it's watching a movie. It knows if a cell is shrinking right now or if an angle is opening up right now.
So the computer has this massive data set, geometry plus time. How does it even begin to process it? This is the message passing part. But don't just tell me it learns. How does the math actually move?
Think of it as iterative updating. This uses what's called a graph transformer. So in step 1, a cell looks at its own data. I am shrinking.
Okay.
In step 2, it aggregates the data from its immediate neighbors. It doesn't just copy them, it runs a function to weigh their importance.
So if my neighbor is under high stress, that message gets passed to me.
Exactly. And in the next layer, you receive the aggregated messages from your neighbor's neighbors. Information literally flows across the graph.
I see.
The AI aggregates this local geometry into a massive high-dimensional hidden state. So by the end of the process, every single cell has a complex mathematical signature that represents not just itself, but its entire context within the tissue.
Okay, that is a dense mechanism. Before we get into the crystal ball capabilities of this thing, let's hit a checkpoint. Checkpoint 1. First, the history. We had the granular view, the particles, and the foam view, the bubbles. Both were incomplete on their own.
Right.
Second, the solution. MultiCell uses a dual graph to map cells and junctions simultaneously.
The houses and the roads.
Third, the data. It captures area, curvature, shear, and crucially, their velocity, or rate of change.
Yep.
And fourth, the processing. Message passing lets every cell feel the stress and state of its neighbors.
Spot on.
So here is a question for you listening right now. Visualize your own neighborhood again. Could you predict a traffic jam just by looking at the driveways of the houses?
No. You'd need to see the intersections, the road width, the speed of the cars.
And that interplay is exactly what the dual graph captures.
And once you have that captured, you can do some pretty amazing things.
Let's talk about that section 2, application.
Yeah.
They use this to solve a massive headache in biology called video alignment.
Right. The global prediction task.
Yeah.
Here is the challenge. Embryos are like bakers baking bread. Even if they use the same recipe, the same DNA one might rise a little faster because the kitchen is warmer. Drosophila fruit fly embryos vary wildly in speed due to temperature or slight genetic differences.
So if you have 2 videos of 2 different embryos, they don't match up in time. 100 minutes in one video isn't the same stage as 100 minutes in the other.
Exactly. It makes comparing them impossible.
And aligning them by hand must be a nightmare.
A total nightmare. So they train MultiCell to look at an embryo and predict developmental time, not clock time, but biological time.
It watches the movie and says, okay, based on the shape of these cells, you are exactly at the gastrulation stage.
And it worked. But here is the magic part. If you look at figure 2 in the paper, they used class activation maps to see what the AI was looking at to make that prediction.
This solves the black box problem. Usually we don't know how an AI thinks, but here they could see which parts of the embryo lit up in the computer's brain.
Exactly. And what they found was startling. The model would light up specific regions, like the ventral furrow or the posterior midgut, before the tissue actually folded.
Wait, so the human eye looks at the video and sees a smooth ball of cells. Nothing is happening yet.
Nothing visible.
But the AI looks at it and says, the ventral furrow is about to buckle right here.
It detects subtle morphodynamic features, micromovements, tiny shifts in shear and angle that are total invisible to the human eye.
So it's predicting the earthquake before the ground shakes.
That's a perfect way to put it. It's reading the pre-tremors of biology.
That is incredible. But that's global, the whole embryo. I want to zoom in. Can it predict what a single cell will do?
This is section 3, predicting the future locally. Specifically, predicting junction loss.
Junction loss. That sounds like a highway exit closing.
Yeah.
What is it in a cell?
In biology, we call it a T1 transition. Imagine 4 cells arranged like 4 squares in a window pane.
Okay.
For the tissue to change shape, to elongate or fold, those cells have to swap neighbors. The vertical line between 2 cells shrinks to a point, vanishes, and then a horizontal line opens up between the other 2 cells.
So junction loss is that specific moment the bond breaks.
Yeah.
That's the mechanism of movement.
Yes. And the researchers asked, can MultiCell look at a static frame and predict exactly which connection will break 1 minute into the future? It achieved 90% accuracy.
90%. That seems incredibly high for a chaotic biological system. Is it really predicting, or is it just memorizing?
That's what they checked. They trained it on one set of embryos and tested it on a completely different embryo where they masked the future frames. So it wasn't memorizing, it was understanding the physics.
But how? What information was the AI actually using? This brings us to my favorite part of the paper, the ablation study.
Yes, the Sherlock Holmes segment.
Exactly.
This is figure 3rd, and it's so crucial for understanding why this works. The researchers started turning off inputs one by one to see what broke the model. First, they asked, is it just the edge length?
That makes intuitive sense, right? If a connection between cells gets really short, common sense says it's probably about to snap.
You would think so. But when they gave the AI only the edge length, the accuracy measured by something called AUC was about 0.82.
Okay, 0.82 is decent, but it's not 90%. It means a lot of short edges don't break.
Exactly. Sometimes they stay short. So then they tried giving it only the cell geometry, the shape of the house, but not the road.
Yeah.
That got an AUC of about 0.86. Better, but still missing something.
So when they combined everything...
So full model hit 0.95 AUC.
So this scientifically proves the initial hypothesis. You cannot separate the granular from the foam.
It proves that the prediction requires the interplay. The stress on the cell body, the area and curvature, combined with the tension on the junction, the edge length and angles, that's what causes the break.
It's not just one thing, it's a system.
The paper describes tissue rearrangement as avalanche-type behavior. It's governed by stress patterns across the neighborhood, not just one local feature.
Avalanche-type behavior.
Oh.
That is a powerful image. Okay, let's hit checkpoint 2.
Let's do it.
First, global alignment. The AI uses activation maps to see events like the ventral furrow forming before they are visible to the eye.
Right, the pre-tremors.
Second, local prediction. It predicts T1 transitions, neighbor swaps, with 90% accuracy.
Yep.
And third, the proof. The ablation study showed that neither cell data nor junction data is enough on its own. You need the dual graph.
Correct.
So quick question for you listening. If you were a cell under pressure, would you crack because you're being squished? That's cell geometry. Or because the bond holding you to your neighbor is weak? That's junction geometry.
The answer is, it's a complex combination of both. And now we have the math to prove it.
So where do we go from here?
Now we need to take this one step further. We've looked at specific predictions, but this model is smarter than just, will this line break? This brings us to section 4, multitasking and latent discovery.
Multitasking. I struggle to walk and chew gum at the same time. What is this model doing?
So they set up the model with a shared encoder, one brain, but gave it 3 different output heads. Basically 3 different mouths to speak 3 different predictions at once.
And what were the predictions?
It had to look at the tissue and simultaneously predict the time until 1, invagination or folding in, 2, division, which is mitosis, splitting in 2, and 3, rearrangement, the neighbor swapping we just talked about.
These are totally different biological processes. Mitosis is about DNA replication. Invagination is about mechanics.
Completely different. Yet the model could predict events 30 minutes into the future with correlations of nearly 0.9. It could distinguish between a cell that was about to divide and a cell that was about to fold.
But here's the part that just blew my mind. The aha moment with unsupervised learning, the UMAP visualization. Explain this to us, because the implications seemed huge.
So inside the AI, there is this latent space. It's the internal mathematical thought process of the computer. The researchers visualized this space as a 2D plot.
You're right.
Now remember, they never told the model which part of the embryo was the head, the tail, the dorsal side, or the ventral side. No labels whatsoever.
It's flying blind regarding anatomy, just these shapes.
Exactly. But when they plotted the latent space, the cells naturally separated into clusters. And those clusters perfectly matched the real biological domains.
Wait, hold on.
The AI put all the head cells in one pile and all the tail cells in another.
Purely by looking at the geometry, the shape of the hexagons and the angles, the AI figured out, I am a head cell, or I am a stomach cell.
Precisely. The implication is profound. Structure dictates function. The geometry of the cells is their identity. The model discovered tissue anatomy just by doing the math of shape.
That is wild. It implies that if you look closely enough at the shape of a cell, you know its destiny. There is no such thing as a generic cell.
In many ways, yes. The history of the cell is written in its geometry.
So where do we go from here? Section 5. Broader implications. Why does this matter for things other than fruit flies?
One major area is connecting live and fixed data. This is a huge problem in biology.
Okay.
Omics, genetics, protein analysis, usually requires you to kill the cell to analyze it. We call that fixing the sample.
So you can see the chemistry, but the movie stops.
Right. And live imaging shows the movie, the movement, but you can't see the chemicals. MultiCell acts as a bridge. Because it understands the geometry so well, you can use it to align the live videos with the fixed samples.
So you can create a 4D morphodynamic atlas.
That's the goal.
It's like watching a silent movie, the movement, and then using AI to perfectly dub the sound, the chemistry, back onto it to get the full picture.
A perfect metaphor. And this leads directly to disease modeling. Think about what we just discussed. Prediction of rearrangement. Many diseases, kidney cysts, spina bifida, cancer metastasis, are caused by dysregulated rearrangement.
Cells moving when they shouldn't or not moving when they should.
Exactly. So if you have this model, you can perform in silico drug screening.
In the computer.
You could simulate a drug, see how it changes the cell geometry in the computer, and the multi-cell model could tell you, hey, this change in geometry is going to cause a cyst to form in 30 minutes.
You can test the side effects on tissue architecture without ever touching a patient.
That is the dream. And finally, the paper mentioned evolution.
Right.
They suggest applying this to archaea or ichthyosporia organisms that aren't quite animals but form colonies.
So are they asking if physics is universal?
Exactly. Do the cells of a primitive colony obey the same geometric laws as a fruit fly or a human? MultiCell provides the ruler to measure that. It lets us ask if the rules of life are actually just the rules of physics.
Heavy stuff. Okay, let's do our final checkpoint 3.
Bullet summary time.
1. Multitasking. The model can predict division, folding, and swapping all at once.
2. Latent knowledge. The AI discovered anatomy, head versus tail, purely from geometry, no labels needed.
And 3. The future. Merging this with genetics to study cancer, birth defects, and even evolution.
Got it.
Incredible. So bringing it all home, what is the big takeaway?
The big takeaway is unification. We spent a century arguing if tissue was particles or foam. MultiCell proves that both were right, but incomplete.
Right.
By combining them into a dual graft, we can predict the behavior of living matter at single-cell resolution.
It's like we finally found the Rosetta Stone for cell language. It just turns out the language is geometry.
And dynamics. Don't forget the rate of change.
Right. Geometry in motion.
Yeah.
So here's my final thought. It's like predicting the traffic flow of a city. Not by reading the minds of the drivers,
No.
but by understanding the architecture of every single house and the angle of every single intersection simultaneously.
And realizing that the architecture forces the traffic flow. The shape guides the behavior.
Which leads to a provocative question for you, the listener. This model predicts cell behavior based solely on geometry and neighbors. It doesn't know about genes or proteins directly. So, if our cells are just following complex geometric rules, how much of biological design is actually just physics taking its course? Are we built by a blueprint, or are we just the inevitable result of bubbles trying to get comfortable?
That is a question that will keep you up at night.
So tonight, when you are doing the dishes, look at the bubbles in your soap. Watch how they pop and rearrange. They are obeying the same geometric laws as the cells that made you.
Physics is everywhere.
Stay curious and keep diving deep.